This is Unit 8, Part 6. Lipids covers quite a wide variety of molecules. When we talk about fats in the diet, typically we're talking about triglycerides or neutral fats, and that's what we're going to be discussing here. So here is the food stuff. And this is the enzyme that acts on it and the source of the enzyme, the site of where the action is occurring, and the path of absorption, which in the case of fats is a little more complicated than with amino acids from proteins or monosaccharides from polysaccharides. Well, first of all, you should, as I mentioned in a previous unit, you should realize, of course, that fats tend to conglomerate together. They adhere to each other if they're in an aqueous solution. They are polar and hydrophobic. You pour some oil on top of water and shake it up, you'll have it mixed up reasonably well, but it quickly separates with the fat usually floating on top and the water underneath. So fats need to be emulsified. Emulsification refers to making it possible for them to be in tiny droplets. Once they're emulsified, then enzymes can act on them, because then there's a much bigger surface area with all those millions of tiny droplets, as opposed to one large layer of fat which the enzyme can barely act at the surface of. So that is what bile does. The bile salts in the bile act to emulsify the fats in the diet. So bile salts come down the bile duct from the liver, well, from the gallbladder where they were stored after being made in the liver, and enter into the small intestine and act on the fat that's in the meal. And I would remind you that one of the triggers for that would be having a fatty meal and the release of hormones like cholecystokinin. So once it's emulsified, then the pancreatic lipase, the pancreas produces an enzyme called lipase. Then pancreatic lipase, which also comes in from the pancreas and also goes into the small intestine, then it can act on the emulsified fats and split them into either glycerol and fatty acids, or sometimes you get two fatty acids cleaved off of the fat molecule, the triglyceride, and you get a monoglyceride. That's a glycerol with one fatty acid chain, and fatty acids, the other two that were released. Now the fatty acids and monoglycerides, they enter the intestinal cells by diffusing across into the intestinal cell. Okay, they can pass across the cell membrane, so they go into the cell that lines the surface of the gut mucosa, which of course are the columnar epithelial cells. There they combine with proteins inside the cell and become what are called chylomicrons. The chylomicrons then can pass into the lamina propria and enter into the lacteals and get transported into through that lymphatic system, which eventually will join with the blood vasculature. All of the lymphatic vessels join up as they move up the arm and up the body, up the legs, and get into larger lymphatic vessels until they finally empty into the blood via actually there's two entry points, but the main one is the thoracic duct. Glycerol, unlike fatty acids and monoglycerides, glycerol and short-chain fatty acids, notice it's short fatty acids, the other one would be longer chain fatty acids. So glycerol and short fatty acids can be absorbed right into the capillary blood in the villi, just like the monosaccharides from polysaccharides and the amino acids from protein. And they get transported through that blood first back to the liver along with all the venous return from the abdominal organs, and they enter into the liver via the hepatic portal vein. So let's go through what I just said again. Triglycerides, or neutral fats, and their breakdown products for that matter, are insoluble in water. They won't go into solution. They'll stay in a separate phase. They will tend to group together because they are hydrophobic. They need to be pre-treated in order to get digested by chemical digestion and absorbed. The small intestine is the only site where fat digestion occurs, and the pre-treatment required is the addition of bile salts which can emulsify the fat. So the fat needs to get emulsified, and it's done by the action of the bile salts. Now, the bile salts, and here's a diagram of the bile salt, it's actually very uncomplicated, simple how they work. Bile salts have both a polar and a non-polar region. There's the polar regions marked, the non-polar regions. This makes them amphipathic molecules. And they will attach via the nonpolar region onto the surface of fat droplets. In that way, you can get many, many small fat droplets. In other words, in that way, the fat can be emulsified. And now you have many droplets coated with bile salts. The polar regions are facing outwards in contact with the water, which is also polar. And the non-polar regions are facing inwards to the oil, which is non-polar, just like they are. Now, lipase can act efficiently on this lipid because of the much bigger surface area now that they're small droplets. So lipase, which is secreted by the pancreas into the small intestine, is able to cleave off two fatty acid chains from the triglyceride to form monoglyceride and glycerol molecules. To form monoglyceride and some free fatty acid chains. So essentially what it does is it can cleave off two. Okay, so it cleaves off, and these are separate, and then the monoglyceride is left. Yes, here I have it here. Two free fatty acid chains and one monoglyceride is the result. Now, that's not the end of it. What happens is that the fatty acids and the monoglycerides get absorbed. They go into the intestinal cell via diffusion. So now they're in the cytoplasm of the intestinal cell, the surface of the mucosa, the simple columnar epithelial cells, and they combine with proteins inside and resulting in chylomicrons, which then enter the lacteal and go via the lymph. They get into the blood circulation. The glycerol and short fatty acid chains that are left absorbed. It's a repeat of the slide with the boxes. Absorbed in the capillary blood, go into the liver via the hepatic portal vein. Remember, bile salts are not an enzyme. They don't have enzymatic activity. Pancreatic lipase is an enzyme, but bile salts are not an enzyme. They are a product of the liver. In fact, the liver doesn't produce any enzymes. Here we have a more detailed description of what's going on, what's happening in the digestion, absorption of fat. You have chylomicrons, you have emulsification of fat, and these emulsified droplets are called micelles. So you have fatty acids and monoglycerides in the form of micelles, and the lipase acts on them. That's has acted on these droplets to cleave off the fatty acids and monoglycerides from the triglyceride. The fatty acids and the monoglycerides go across into the columnar epithelial cells covering the villi. They enter by diffusion, and they're used to synthesize triglycerides again inside this cell. So the triglycerides reform inside the endoplasmic reticulum. These form small globules associated with proteins, which are now, and that combined with proteins, the fatty globules combined with proteins are now called chylomicrons. So you go from fat to emulsified fat to fatty acids and monoglycerides associated together into micelles, into the cell to form back into triglycerides, and then associating the triglyceride fatty globules that form combined with proteins to form chylomicrons. And then these are extruded onto the basal side. Remember, this is the apical side, and this is the basal side of the simple columnar epithelium. So they go to the basal membrane, and they get extruded out, and they will enter into the lacteal, modified, specialized lymphatic capillary in the center of the villus. And the lymph in the lacteal will transport those chylomicrons away from the intestine through the lymphatic vascular system up to a point in the chest where they enter into the bloodstream. Just want to remind you that the bloodstream, and this is just, a simple diagram of it, has a pump. The cardiovascular system has a pump, the heart. It pumps oxygenated blood out to the tissues, goes into capillary beds, and then goes into the venous side and returns to the heart. Meanwhile, throughout your body, there are also lymphatic vessels, tiny little lymphatic vessels. We're talking here about specialized ones in the villi, but it doesn't matter. They're all over the place, and they drain any fluid that in the extracellular space that's outside of cells, outside of the vasculature, so in between cells. And some of the fluid from the blood actually goes into that space. Anyhow, it all drains back, and it goes into larger and larger ones until it finally goes back into the rejoins the blood vasculature. Along the way, you have these little filters called lymph nodes. And here it shows a diagram of how closely interconnected these lymphatics are, very in close association with the capillary beds. Here you see this, and just a very small part of the lymphatic vasculature. If they showed everything on this, it would be one solid black, you wouldn't see any detail at all. So they're just showing a few, and they're showing that these drain upwards, and they go into the chest in two spots, here and here. There's more flowing in on the on the left-hand side, on this side, the left-hand side of this person, through the thoracic duct, which is located right there. right into the thoracic duct there. That's the point where it goes in. This is the right side. Obviously, this is the right side of the person, and on the right side you have drainage just from the arm and from the right side of the head and maybe the right upper chest, draining, lymphatic drainage going into the right subclavian vein. Here it goes through the thoracic duct into the left subclavian. So that's how fat is moved around, absorbed, first emulsified, then diffuses across, reforms, first from micelles, then into chylomicrons, then into fat, and then into chylomicrons. It first is emulsified, then broken down by lipase, then diffuses in, reforms into triglycerides, and then gets carried across and is now called chylomicrons, and then extruded across, goes into the villi, the lacteal of the villi. Okay, let's talk about the fourth type of major molecule found in living things and in the diet. Everything eats living things, things that were once living. So the fourth main molecule are nucleic acids. This shows a simple diagram of the structure of DNA. DNA is a multi-strand, polymer made of many building blocks. The building blocks are nucleotides. There are two strands. DNA is a double-stranded molecule. Each strand is a single polymer, and they wind around each other, sort of like this. Let me draw the other one in a different color so you see. They wind around each other, and they can be much longer than this. What you're seeing, and the rest of the molecule is located across, just like sort of like steps in a ladder. You have it going across like that, and that's what's shown here. They're just showing one. In this diagram, they're showing phosphate group and a monosaccharide. In this case, it's DNA, so it's deoxyribose, and that would be located, let me just get a good color for this. So those would be located here. The phosphate and the deoxyribose would be located there. And then the nitrogen, notice that this has nitrogen in it. These contain nitrogen here. These are nitrogen containing bases, part of the nucleotide. So the nitrogen containing bases would be located across here. So what you see here are two nucleotides with one nitrogen containing base there and a nitrogen. A nucleotide is made of three parts: the nitrogen containing base, the deoxyribose monosaccharide or sugar, and the phosphate. And similarly on the other side, you have the nitrogen containing base, the deoxyribose, sugar, and the phosphate. So these are two nucleotides which associate with each other by hydrogen bonding. Hydrogen bonding is shown here and here. Okay, these happen to be A and T, adenine and thymine. Here you see a more detailed diagram with more nucleotides arranged in the polymer form, where you have one strand here, and the other strand of the double helix. They're helically shaped, wrapped around each other, is there, so it's a double-stranded helix. The backbone, which is the strand along the back here, that's what people refer to when they're talking about the backbone. It is made up of strictly the phosphate group and the deoxyribose sugar, and you see the color coding for that. And then the steps in this twisted ladder are made up of these 4 nitrogen-containing bases, A, T, G, and C. If there's an A, stands for adenine, on one polymer strand, then in the equivalent position on the other side, you'll have a T. If there's a T on the other side, you'll have an A. So wherever there's an A, there's a T. Wherever there's a, and vice versa, T, A, T, they will associate with each other through hydrogen bonding. And wherever there's a G, there will be a C on the other side. Let's do that in a different color. Wherever there's a G, as you can see here, there'll be a C. So GC pairs everywhere. Always GC pairs. ATGC, that's all you have to remember, ATGC. Notice that there are 3 hydrogen bonds formed between the G and C pairs, and there are only 2 hydrogen bonds formed between the AT pairs. Well, we eat cells, we eat living things. Living things are made of cells, and almost all cells except for red cells have a nucleus, and therefore we are also eating nucleic acids. So we eat nucleic acids, and the enzymes that can break them down are coming from the pancreas. They are ribonuclease and deoxyribonuclease, and also further breakdown by brush border enzymes, nucleosidases and phosphatases. The site of action of these enzymes is in the small intestine. This comes from the pancreas. This comes from the intestine itself. The brush border enzymes, they're coming from little glands in the wall of the intestine. And they act in the intestine to break down nucleic acids, which then get actively transported across the simple columnar epithelium, get absorbed into the bloodstream and the villi, transported to the liver via the hepatic portal vein. So, again, nucleic acids, they get absorbed if they've been broken down into the monomers. Remember, the monomer are the nucleotides, and the polymer is the DNA. They're absorbed via an active transport mechanism involving membrane carriers, and they get transported across and absorbed in the villi and go up through the blood via the hepatic portal vein into the liver. The enzymes involved include what are called generally classified. This is a general term, just like proteases was a general term for many enzymes, including pepsin, trypsin, carboxypeptidase, etc. This is a general term for enzymes that work on nucleic acid. So they include the pancreatic ribonuclease and the pancreatic deoxyribonuclease. And also from brush border, that was from pancreas, also brush border enzymes, nucleosidase, and phosphatases. And there you see it. That's what happens. Gets broken down, absorbed in, and transported through the blood to the liver, processed, and then the rest of the blood goes up to the heart through the venous return. Absorption in the small intestine, all nutrients essentially are absorbed in the space of the small intestine. They get acted on by hydrolytic enzymes and absorbed. About 80% of electrolytes present are absorbed, and most, maybe 95% of the water, not all, What remains is some water, some materials that are indigestible, and of course some bacteria. The electrolytes are absorbed. That 80% of the electrolytes that get absorbed, they're actively absorbed. In other words, this kind of absorption needs energy, and the energy source is from ATP. They're absorbed along the whole length of the small intestine. It's a direct active absorption, and also there is an indirect active absorption where sodium is coupled with either glucose transporting in, or sodium gets coupled with amino acid being transported in. Calcium absorption really relates to the blood level of ionic calcium. If the blood level is low, then calcium absorption from the food goes up. If the blood level of calcium is high, then absorption from the gut drops. The overall regulation is by vitamin D and a hormone released from the parathyroid glands, which are in your neck. There are 4 of them, 4 lobes, parathyroid gland. They release parathyroid hormone. When ionic calcium is low, levels are low in the bloodstream, parathyroid hormone release goes up, and absorption goes up, mobilization from the bone. You'll get into this from, we're just talking digestion, absorption, so it's enough for you to know that the absorption will go up due to the action of parathyroid hormone. If levels are low in the blood, then less parathyroid hormone is released and less absorption is occurring. So it all is related to blood levels of ionic calcium. Iron absorption is a bit different. The body does not give up iron at all. Any iron in the diet will be absorbed, to the extent that if you happen to have too much iron in your diet, you will keep absorbing and you'll have iron overload. Iron physiology is fairly complex. We're not going to get into hardly any of it. Enough to say that ionic iron gets actively transported into the mucosal cells, where it binds to a molecule called ferritin. Ionic iron is actually fairly toxic, so it has to get bound up once it's in the body. It binds to ferritin. Vitamins are important molecules. They're not all grouped under vitamins because they have similarities in their structure. In fact, for the most part, none of them have any similarity in their structure. They were identified as key types of molecules which had to be absorbed and had to be obtained in the diet or from the gut in order for a person to remain healthy. And that's how they got their name, including the term vita, which derives from a Latin word for life. The absorption of the vitamins depends on whether they are fat-soluble or water-soluble. The fat-soluble vitamins that we need and absorb are A, D, E, and K. And to make it easier to memorize or remember, I just go with ADEC. So these combine with me cells. You'll remember me cells that are formed in the gut and absorbed passively. Among them, there's one that's interesting because it's not in the diet. It is actually made by bacteria in the gut. Enteric bacteria. Enteric refers to intestinal. So enteric bacteria synthesize vitamin K, and then we absorb it. Water-soluble vitamins include vitamin B and C. They're absorbed by diffusion. There are several different vitamin Bs. There is one which is unusual. It is vitamin 12. It is absorbed, always bound to a molecule made in the stomach, which we've referred to before, called intrinsic factor. It gets endocytosed across. Water absorption, as I said, 95% in the small intestine. That is movement across a semi-permeable diffusion across the semi-permeable membrane, in other words, osmosis, and the remainder, almost every last drop of it, not quite every last drop, but the vast majority of the remaining 5% gets absorbed in the large intestine. It's important to understand that water can actually move either way. It could also move in. If you have a high electrolyte level in your gut, it could move across from your tissues into the space of the intestine if there's a hypertonic solution in your intestine. This is the last slide. It involves malabsorption of nutrients. And I can tell you that, it's a big topic. We're keeping it simple, but anything that interferes with delivery of either bile juice, remember not enzymes, but this mostly it contains, the important key thing is bile salts, and pancreatic juice, which is several different enzymes. We've now discussed quite a few that you need to know. So anything that interferes with that will interfere with absorption, because these are key. These molecules are key to properly processing and absorbing either fat, in the case of bile, or nucleic acids, carbohydrates, or proteins, in the case of pancreatic enzymes. Anything that damages the intestinal mucosa, which can happen with certain bacterial infections, and is not uncommon. Almost everyone occasionally gets some sort of bacterial infection that causes diarrhea, diarrhea really is a reversal in the direction of which way the water is going, because bacteria often will produce toxins that can cause ions to move from the tissues into the space of the intestine, and that'll pull water with it, and with the increased amount of water you end up with diarrhea. A lot of people now are interested in gluten. Gluten happens to be a protein in certain associated with certain starch products, I guess, but all sorts of people think that they have to have a gluten-free diet. There's only a small percentage of the population which has a particular autoantibody, an antibody against self molecules, which it's only those people who have to have a gluten-free diet. They're the only ones who could develop gluten enteropathy, disease of the intestine. That disease is celiac disease, but it's only those people with that autoantibody. If you don't have the autoantibody, you don't have the sensitivity, you don't have the disease, and you're wasting effort in avoiding gluten. Problem with gluten is you get damage to the intestinal villi and the length of the microvilli. The treatment is avoiding gluten in the diet. All grains but rice and corn. So these people have to avoid all grain products, but rice and corn are okay. And that's the end of Unit 8, Part 6.